Next speaker is Dr. Matthew Breen, who's a distinguished professor of comparative oncology genetics at North Carolina State.
Thanks, Timmy. There we are. I'd like to thank the organisers for asking me to come and talk to you briefly today, although I'm afraid asking me to speak briefly is like throwing a tennis ball for a golden retriever and saying, sit. But let's see how we get along. So I was asked by the organisers to talk a little bit about the importance of having reliable biobanks available for canine tumour studies, and then to talk a little bit about how we're using some of these biobanks, wherever they may be, and however good or not good they are, in terms of progressing what we know about the comparative aspects of canine oncology. But this is the whole concept of comparative oncology stems from the one medicine concept. And as you can see from this slide, this is nothing new. This has been around for over 100 years. It's a concept that, with no disrespect to the MDs in the room, MDs treat one patient and the DVMs treat everything else that moves. There is an opportunity to take advantage of this by merging it all together and then applying this to cancer research. The traditional cancer model is, of course, the mouse. And I think, as Dr. Kanna eloquently stated, I don't think any of us that work in the comparative oncology community have any problem with the mouse. The dog as a species, as with other animals on the planet, really are complementary additive species to kind of add to the totality of the approach. It's not a threat to the mouse, it's an addition to the mouse. And for that reason, in my lab at North Carolina State, we work on cancers in lots and lots of different species. And I could spend 20, 30, 40 minutes talking about every single one of these. But the main thing that really strikes home is that all these cancers are spontaneous. And this is really an important point to drive home, because many people get confused when we talk about using the dog as a cancer model, in that they say, how could you cause a dog to get cancer? And I think what really needs to be made very, very clear is the dogs that we work with, as with all the animals we work with, they are spontaneous diseases. They are not induced, they are our pets. They live in our homes. They breathe the same air, they drink the same water, sometimes they eat the same food. And when you throw a ball across the park, your dog runs across the same herbicide, insecticide, pesticide-treated grass that you, your children, and your grandchildren run across. So it's actually no major surprise that if there are environmental influences affecting cancers, those kind of environmental agents are affecting pet animals as well as human beings. But of course, we don't have time to talk about all these species. We only have time to talk about the dog. And when we think about the domestic dog, as with all animals, really what I'm trying to convince you to go home with is the thought that really animals are just differential rearrangements of the same collection of ancestrally related genes. Shuffle the deck and you're a human being. Shuffle the deck differently and you're a dog. Shuffle the deck a third time and you're a cat. Think about that in the back of your mind. But when we think about dogs in particular, we know that cancer is a genetic disease. We know, and we can talk about this till the cows come home, that there are certain breeds of dogs that are very, very highly predisposed to certain types of cancers, which tells us that there is an inherited predisposition, which plays a role in reducing the level of genetic heterogeneity through descendants. And as I've pointed out, it's the same environment. So when we sit here thinking about the powerful role that a dog can play, think about it as the opportunity that we now have today in 2015, with all the tools and reagents that we have, to consider the use of the domestic dog as a powerful opportunity to identify genetic factors that will advance what we know for the purposes of veterinary medicine, but will also augment and potentially enhance what we know about human medicine. So it's no surprising that if we look at the types of different cancers that affect people, the same kinds of cancers in most cases affect dogs. What I want you to think about is not just the one medicine concept, but think about it all as one pathogenesis. And when you look at the numbers at the bottom of this slide, it's approximately 500 per 100,000 people in this country that develop cancer each year. It's very difficult to know what the precise number of dogs that get cancer, but the numbers that I've looked at indicate that the the proportion of dogs that get cancer is probably 10 to 11 times that which we see in people, and therefore again provides a tremendous opportunity. So when we think about dogs getting cancer, therefore why do we need biobanks? What would be the role of a biobank? And I've listed 3 major assets here. Is really what we think about as a dog developing a malignancy is that's a clinical piece of information, and it's a very important clinical piece of information. In order to get to the underlying genetics of it, we really need to bank those biological specimens. But in addition to banking those specimens, we need that clinical data. We need the pathology data that comes with it. We need outcome data. We need to know how to intervene, how to prevent, and how to cure. And really what drives most of the biobanks these days is not just a biobank in North Carolina or a biobank in California. There needs to be shared resources. There has to be a common goal with a common vision. Sharing the data as well as the samples is fundamentally important to performing the benefits of the role that we're trying to play. So when we think about what makes an effective biobank, and I've just listed a few points here, we really need what are called standard operating procedures. It's the same with any collection that is going to be accessed by multiple people. We need very, very high quality specimen selection and storage. Those specimens need to be fit for the purpose that they are destined. There's no point in taking a tumor tissue, leaving it in a veterinary clinic for 6 months, and then deciding to snap freeze it. There needs to be high quality preparation, high quality storage, and above all, an easy and rapid means to retrieve those samples from a bank. When we think about specimen access, not just the specimens need to be available, there needs to be access to the patient information, the pathology, the outcome data that coincides with those particular patients. And of course, it's all well for us to have our own individual proprietary banks, but what's really needed is a data sharing plan. We need to have a means whereby which people can access data to help them select the most appropriate cases for their own research enterprise. So there are now several biobanks around the world with an emphasis on canine specimens. I'm just going to talk primarily about one, but before I do that, I want to come back to a question that was asked in one of the previous sessions, which is the one on the right-hand side, is the Morris Animal Foundation's Golden Retriever Lifetime Study, which is a collection of 3,000 golden retrievers. And the really nice thing about this biobank is it's specific to one breed, and one breed so far that is incredibly popular in the US. And these dogs are being sampled every single year during their lifespan from age below 2 right up until through the time that they die. And a whole suite of different biological specimens are being collected from them, including an enormous amount of patient information and client information about where those dogs live, what kind of water those dogs eat, what kind of food those dogs eat. The environmental factors that come with this should not be dismissed. What I really want to talk about in terms of biobanking is the thing that Dr. Kanner alluded to, which is the Canine Comparative Oncology and Genomics Consortium. And this is a kind of a community-driven enterprise by the community for the community, which was set up several years ago with seed money that were received with a $1.1 million gift from Pfizer, and then $0.6 million from the American Kennel Club's Canine Health Foundation, and $500,000 from Morris Animal Foundation. And with that $1.7 million over the ensuing several years, we've put together a detailed physical infrastructure. We've developed a database and how to manage that, and so far have collected slightly over 2,000 patient samples. But importantly, those samples all come with high quality and assurance. When this originally was set up, we identified 8 different collection sites, and those collection sites represent some of the nation's leading veterinary academic medical institutions, 6 of which are currently still active. And those sites, the key activity is they were involved in helping to develop the standard operating procedures. The sites signed up for helping to design the best way in which these samples should be collected, and then strictly adhered to those SOPs during the collection process. The presence of the Pfizer CCOGC biospecimens repository is really there as a consequence of that very rigorous standard operating procedure. What happens is samples are collected from each patient, and think about it, as I say here, it's one patient, but because of the diversity of samples that are collected from each cancer-bearing patient, there are many opportunities for downstream research, whether it's veterinary-oriented or comparative-oriented. And then one of the things that I really want to make very clear is this is really an American citizen enterprise. Every single one of the dogs that is in part of the CCOGC's biobank is somebody's pet. And that is actually incredibly important, because I never cease to be amazed of the courage that animal pet owners have when their dog is diagnosed with a neoplasm or potentially is entering the clinic to be put to sleep. Those owners have the courage to rise above their own personal trauma and say, I know I can't do anything for my dog, but I am going to allow my dog to have biological samples taken, because those samples may benefit the next generation of dogs. So when I say it's a collaboration with pet owners, who often referred to be called pet parents, the relationship between a dog and their owner is actually very, very strong. It's a very strong bond. It really is an academic, citizen-based collaboration. Whether those same people would be interested in providing those samples if it was purely for a for-profit enterprise is another question that would have to be debated later. But as of now, people provide samples from their dogs because they care and they want to make a difference. In the CCOGC's biobank, we now have over 60,000 biological specimens representing almost 2,000 patients. And I've just listed here the different types of samples that are available for anybody who wants to access them for a very reasonable price. This is a not-for-profit. This is a perpetuity model. The cost of the samples is just enough to cover the cost of replacing them. But more importantly, the bank has gone through a very rigorous QC process. The QC process was presented at the AACR meeting in 2013. And what came out from that particular study is that as part of a pathology review that was done by people at the NCI, 331 cases were looked at just to make sure that the piece of tissue that is in the tube is what it's supposed to be, because that in of itself could cause potential problems. And as you can see from this list, most of the cancers that are there have a very, very high correlation in terms of what's supposed to be in the tube and what's not supposed to be in the tube. And the only low one would be something like an hemangiosarcoma, and that's because the tissue in the tube may not be highly enriched for neoplastic tissue or may be more of a hematoma than an actual mass. That's a problem that clinicians face, pathologists face all the time. Taking it a step further, the pathologists at NCI then looked at 132 cases and actually went further and said, in addition to how accurate was the diagnosis, how pure is the tumor specimen? And this comes back to what Debbie spoke about a minute ago about heterogeneity. This is a problem. If you start looking at a whole 100 different tumor tissues and they have incredibly varying levels of heterogeneity, are you really looking at the same cancer specimen? That needs to be taken into consideration. So the CCOGC spent a lot of time and well over $100,000 actually making sure that the specimens in the bank are catalogued according to their level of heterogeneity. And to take it even one step further, we then looked at nucleic acid QC. If you look at many human tumor banks, they don't do this, and often the problem is that the quality of the nucleic acid, especially the RNA, can tend to be somewhat limiting. So what we did was selected almost 200 cases that broadly represented proportions according to the number of tumors in the bank, but also to test the SOPs by screening across the collection sites, so equally distributed. And I'm actually delighted to say that, I'm not surprised, but I'm delighted to say that the quality of the tumor DNA and the tumor RNA is actually pretty remarkable. And I don't think I've seen this level of quality in most of the human banks that I've looked at previously. But one thing that it also raises is the importance of pathologic review. So this is a slide that shows a biopsy of an inflammatory mass keeping the theme of bladder cancer with a Canine TCC. And if we just take a section from the full size of this piece of tissue and then profile it for DNA copy number, as you can see on this image here, it's flat. It's also very noisy. But if we use a pathologist to go in and say, okay, identify the region of enriched neoplasia, isolate that region from the slide, and then profile it for DNA copy number, the situation is now completely different. Taking it a step further, we've just spoken about BRAF mutations. We independently discovered a BRAF mutation using exome analysis and have developed an assay that allows us to detect a frequency as low as 0.01% of cells. In this particular case here, if you take the full section of the slide, the BRAF frequency is 1.2%. If you actually take the enriched neoplastic region, it's now 12.2%, which goes to demonstrate that don't think that samples that come out of banks are the end of the story. They need rigorous pathology intervention. So how are we using that in my lab? So I'm very quickly going to go through some of the work that we've done to take cancer patient specimens and use them to benefit both cancer research in dogs and people. Now, we can go through this entire list and talk about the many, many different ways that we have used the model. My background is in molecular cytogenetics. I'm a chromosome biologist, so I'm going to focus on the role that we've used, the information we've seen where the dog is used to look at somatic changes using the big picture, as in chromosome changes. This picture on the left-hand side shows a typical metaphase preparation of a human patient. This happens to be male because the little chromosome in the middle is a Y chromosome. And then on the right-hand side is a typical mid-metaphase preparation of a dog. And I'm sure you can all see the driving home message is dogs have lots more chromosomes and they're actually quite a lot smaller. The total amount of DNA is actually not that different. It's just packaged in a different way. So if you look at the generic cartoons, what we call ideograms of how human and dog chromosomes are drawn, this is how we represent them. Now, that's fine when the chromosomes are normal, but during cancers, there is an awful lot of genomic reorganization that occurs. And these little cartoons just demonstrate some of the types of genome shuffling, gains, deletions, recombinations that can occur, which may be a cause of tumor initiation and or progression, or maybe completely coincidental. So the question is, how do we identify in human patients, as well as dog patients, which of these changes are significant, which of these changes could be associated with response to a therapy, and which are just coincidental. So we started doing this maybe 10 years ago and asked this question: are there evolutionarily related chromosome aberrations in dogs that suggest an ancestral mechanism of pathogenetically significant events by the fact that they're shared with people? So to do that, we looked at a whole series of well-described human cytogenetic changes that drive therapy and identify whether they are present in the corresponding cancers in dogs. And I'm going to give you one example, but it's one example of many, and it's also probably the most famous example, which is the Philadelphia chromosome, which is an aberrant translocation between human chromosome 9 and human chromosome 22 that results in the genes BCR-enable becoming co-localized on a derivative chromosome that is called the Philadelphia chromosome. It's a tyrosine kinase, and it's a particular characteristic feature of chronic myeloid leukemias. It's also where the drug Gleevec stemmed from in terms of what Time magazine called the magic bullet. That's one of the success stories. People with chronic myeloid leukemia may be treated with Gleevec, and they actually tend to respond very well and go into what we call cytogenetic remission. And this is an example of what the data look like in a cytogenetic experiment. So what we predicted is that if dogs have CML, and unfortunately the diagnosis of CML in dogs from a research perspective, it's probably as rare as rocking horse manure. They are not very common at all, or maybe they are underdiagnosed, but we have probably recruited maybe 35 to 40 patients in the last few years. So what we've done is isolate the genes BCR-enable, map them onto dog chromosomes you can see here, and then we predicted where that translocation event should be if indeed dogs have a BCR-able translocation. Now, if it didn't happen, I wouldn't be showing it to you, but it actually does. So what you can see here is a BCR-able translocation in a chromosome that's a derivative 26. These patients, several of them have now been treated with tyrosine kinase inhibitors, and what we start to see is a decline in the number of BCR-able positive cells. Whether that has an impact on longevity, we don't yet know, but it demonstrates that despite 60, 70 million years of evolution, dogs and people have a corresponding chromosome aberration from genes that are dispersed throughout the genome. I can show you many, many, many examples of that. But what we're really interested in is, what does that mean? What it means for the dog patients is that we can start to think about targeted therapies based upon targeted genome changes in dogs. Well, that's great. But what we're really interested in as well is, can we actually use the dogs to benefit people. In a comparative oncology, it's a two-way street. So the question we wanted to really ask is, can we use the genome of the dog to identify genes in humans that have thus far not been discovered looking at people alone? And to do that, I want to come back to the statement that I made earlier, which is stop thinking about people as people and dogs as dogs, and think about them as this ancestrally derived form of genes. So these are the two ideograms that I showed you earlier. Now this is just the human ideogram, but onto the human ideogram, as a consequence of the genome sequencing project, I'm now going to overlay all what we call the evolutionarily conserved chromosome segments. It's a bit of a muddle, but there are about 215 of them. So don't think about the human genome now as 22 autosomes plus an X and a Y. Think about it in relation to the dog as 215 differentially sized fragments. How does it work? Let's pick on the baby chromosome 22. Human chromosome 22, according to this chart, is evolutionarily conserved with 3 little regions of 3 dog chromosomes. Those regions are highlighted here. So in a very naive sense, if I took a pair of scissors and I cut out the region marked in blue here, the region marked in green here, and the region marked in red here, and then glued them back together again, I've effectively made the dog equivalent of human chromosome 22. So what? How does that help? Well, it helps because using this differential organization acts as a filtering mechanism. And for that example, I'm going to show you a quick set of data that actually comes back from 2009 through a collaboration that was with NC State and researchers at UC Davis, where we looked at meningiomas. Now, human meningiomas tend to have a deletion of some or all of human chromosome 22. That chromosome is 50 million bases long and has about 500 genes. By looking at meningiomas in dogs, we've actually been able to identify that the evolutionarily conserved region is only about 2 megabases in size and contains fewer than 10 genes. So if indeed these two cancers are caused by the same genetic events, we've now shrunk that region of interest from 50 megabases to 2 megabases. But more importantly, the number of genes that are shared is fewer than 10. And over the past few years, we're now looking at the role that those genes play in human meningioma patients. And the data so far are becoming quite intriguing. One more example, just to keep in line with the canine bladder cancer theme, is when we think about canine bladder cancer in the veterinary space, this is a difficult cancer to diagnose. There may only be, and the numbers again are variable, 1 to 2% of dog cancers may be transitional cell carcinoma, but there are well over 2.5 million dogs each year that are diagnosed with urinary tract issues that could relate to a bladder cancer. So when I was speaking to the veterinary community a couple of years ago, one of the things that they absolutely asked for was, why can't we develop a free catch urine assay that allows us to identify those dogs that have a bladder cancer in the background of an infection? So we actually perform whole genome profiling on a relatively small cohort of canine bladder cancers, and were amazed and astonished by the results, in that the frequency of 3 chromosome aberrations was remarkably high. By using those 3 chromosome aberrations in combination, we have now developed a cytogenetic assay, a bit like using HER2 in breast cancer, that allows us with more than 99.9% sensitivity and specificity to identify the presence of a canine transitional epithelial malignancy in dogs that are symptomatic. It's also now being used to screen early presence bladder cancer in dogs that are not symptomatic. But that's good for dogs. How does that benefit people? How it benefits people comes from a concept we call genomic recoding. And this is where we stop thinking about dogs as dogs. We stop thinking about people as people. We now think about everything in the context of the human genome. So what we did is we actually took all the canine data. These are the data again presented as dog. But now take the genome coordinates and convert them in an output format that represents the human genome. So now these are dog data recoded as human data. And what that allows us to do is to now merge the human data from human urothelial carcinoma patients with our dog data with urothelial carcinoma patients. And what we find is that dog chromosome 13 splits across 2 chromosomes, dog chromosome 19 splits across 2 chromosomes, dog chromosome 36 goes to dog chromosome 2. But when we compare those data to the corresponding malignancies in human patients, now it starts to get very interesting. Because what happens now is we can take a chromosome that's looked at in urothelial carcinomas in people, and you'll see that these two pictures here look remarkably similar. These are dog data as human, these are human data as human, and they look pretty comparable. However, if you look at chromosome 8, for instance, one of the common aberrations in human urothelial carcinomas is an increase in the long arm of chromosome 8. This is this blue region here. However, if we use the dog as a filter, what we can say is, well, if the dog and human diseases are pathogenically related, maybe we don't worry about the proximal half of of human chromosome 8q, just focus on the distal half. That works very well and actually reduces the gene list when we start incorporating other species to one gene. And that one gene is now being intensively investigated in a whole series of human urothelial carcinoma patients. So to summarize, what we've done is we look at a whole range of different cancers. We've taken exactly the same process and looked through, this is a slide from a few months ago, so this is now over 1,500 different patients. And in every single time we've done this, we have been able to identify regions of the dog genome that show evolutionarily conserved chromosome aberrations in people and are focusing that down. Pathologically, this is a group of 60 canine malignancies. The pathologist told us, Matthew, these are all exactly the same thing. Clearly, as we know from human studies, as in dog studies, molecular profiling starts to add value to what we cannot see through a ×40 objective. But interestingly, it gives us the opportunity to ask questions. These data clearly break into 4 groups. We can then ask these 2 questions. Do any of these recurrent genome aberrations actually correlate with prognosis? Or if we can twist the arm of a pathologist, do they look as though they could subtype? And importantly, are there corresponding cytogenetic lesions in human patients? The answer to both those questions is an uncategorical yes. So the dog is actually starting to drive biomarker discovery that is then being applied to human patients. We take a whole, what I call 360-degree approach, and by looking at animal cancers first, and then constantly interacting with our human medical colleagues, exchanging information, acting both as a cross-filter, working with pathologists, working with epidemiologists, and working with clinicians, we have an opportunity not just to develop new diagnostic and prognostic signatures for dogs, but also for people. So I'm going to leave you with this penultimate slide. Is this really a time to hold hands between the human oncology field and the veterinary oncology field? Both human oncologists and veterinary oncologists are trying to do the same thing. But I do believe that when we think about the genetics of purebred dogs and the opportunities we've got with dogs in general, the keys to unlocking some of nature's most intriguing puzzles about cancer may actually be walking right beside you when you go out for your morning run every morning. And with that in mind, and today's a great day to tell you this, is that the Royal Society today has just published an issue on comparative oncology, which is available online today, which talks about the benefits of comparativology and Peto's paradox. So I'd encourage you all to go and take a look at that. It takes a lot of people to do this kind of work, many of whom are in this room. So I want to say particular thanks to the people in my lab, the other faculty we have at NC State, who are a tremendously talented group of clinician scientists, epidemiologists and biostatisticians, and then a whole host of collaborators from across the world, as well as the people that fund this. But I also want to say a big thank you to the thousands, again, of dog parents who have, and their veterinarians, who continue to have faith in the veterinary community to help them solve the problems that their dogs are developing cancers. The only difference is there's billions of dollars each year spent on human oncology and not a significant sum spent on veterinary oncology. I think with the help of this meeting, we have an opportunity to start addressing that as a one species model, not a differential species model. So with that, I ran over a little, but thank you.